Hi, my name is Sithara Vivek. I'm a PhD student at University of Minnesota, majoring in bioinformatics and computational biology. My topic for presentation is variational autoencoder prediction model for Alzheimer's disease dementia. This is part of my PhD thesis project, and I would like to present a proof of concept model I developed. Alzheimer's disease dementia is a complex, heterogeneous neurological disorder, which is a major cause for dementia. As you know, in US, with the aging population, the incidence of dementia is also increasing. So I decided to develop a single omics prediction model and then later develop a multi-omics prediction model for dementia to identify the dementia at early stages. So I used a variational autoencoder, which is a deep learning neural network model that can approximate complex nonlinear transformations to learn the latent representations of this input omics features. I used the data from Health and Retirement Study HRS, which is a biennial survey of older adults over 56 years old in US. And they have collected detailed phenotype and multi-omics data in the 2016 survey. I used the genome-wide epigenetic data to build this prediction model using the cognitive function measures in 2016 survey. The outcome AD dementia is defined based on this cognitive function measures collected in HRS, included episodic memory, mental processing, and working memory. And we got the composite score ranges from 0 to 27. And then I used the Langaware approach to categorize participants into three groups based on the score range to dementia and cognitive impairment, but not dementia and the normal category. For this proof of concept model, I have combined dementia and cognitive impairment, not dementia together to call it as a dementia class. So that we will have enough number of participants to train the model. The DNA methylation profiles were generated by the Illumina Infinium Methylation Epic Bit Chip 850K array in University of Minnesota, which queries the individual CPG sites to estimate the percent methylation rate. As you see in the figure 1, the proposed VAE architecture has a basic VAE model and then a custom classifier network. The basic architecture of VAE includes this probabilistic encoder and a decoder layer with the bottleneck where it represents the latent features learned from the input omics data. And we'll feed the latent vector learned from the input omics data directly to this classifier network for dementia prediction. Since the model has a unsupervised phase, including the encoder and decoder layer, and a supervised classifier network, will use the loss function measured from the VAE model as well as from the classifier loss to estimate the total training error. I've used the hyperparameters with batch size 32 and fixed learning rate at 0.001 and epoch 250 and a latent dimension defined as 128. For the unsupervised phase for the encoder and decoder layers, I defined 3 hidden layers and 2 hidden layers for the classifier network. Among the 850K feature array, we plan to employ various feature selection process based on the variance measured among the features across samples. Also by defining the association of these features with dementia. For the proof of concept model, I have used only the highest variance features, 25K features from the data set. And the class distribution of our data is as shown in the details. Like dementia group included 835 participants and 3183 participants in the cognitive normal category. This proof of concept model accuracy is at 0.69 with a precision score at 0.71 and a recall score of 0.69, which is comparatively lower than the published deep learning model developed using the ADNI dataset to predict dementia. The major reason I think is due to the class imbalance observed in our data set. Also, the features used in the ADNI data set are mainly the classic clinical variables and dementia associated features based on the image and CSF biomarkers. So we hope once we address the class imbalance by proposing to generate more synthetic samples for the dementia category, we will be able to improve the model accuracy. As part of future work, I am to incorporate the existing biological knowledge, for example, gene-gene interaction network as constraints in the model as a regularization step. Also to extract the biological interpretation of latent features by extracting the decoder weights for each input features, and to identify which all are the important input omics features for the prediction of dementia. Later, I would like to develop a multi-omics VAE prediction model for dementia using the GWAS genetic variants and epigenetic and transcriptome data available in HRS. Thank you. I would like to hear your feedback and suggestions. Thanks.